Okay, I guess we'll go ahead and get started. So today we're going to be hearing from Dr. Marcus Foston, and he's going to talk about affordable ethanol, unlocking the secrets to the plant cell wall. So Dr. Foston received his PhD from Georgia Institute of Technology in 2008 and is currently an assistant professor in the Department of Energy, Environmental, and Chemical Engineering here at WashU. His research aims to determine how to efficiently use plant materials or biomass for the generation of biofuels. So we'll learn more about this research today during his talk, and everybody give him a welcome applause.
All right, thank you for that introduction. So, again, my name is Marcus Foston. I'm assistant professor here in the Environmental, and Chemical Engineering department. So I have this lecture, this talk here. But if you have any questions, just raise your hands. I'd rather have a conversation than just sit here and lecture you guys. Um, so in July, I would have been here for 2 years. I teach two courses. One course is called Unit Operations. It's pretty of a standard chemical engineering class. So in a chemical plant, let's say if you're trying to generate a compound C, and the reactants that make C are A and B, you have a bunch of different unit operations. You have to pump A and B around the plant. You have to get it into the reactor. You have to stir it around. You might have to heat it. It might need to be under pressure for a certain amount of time. Once you make C, maybe you make some byproducts. Maybe you then completely convert A and B to C. And so you need to have processes to separate A and B from C. And so the students, chemical engineering students, learn a lot of things about theory. And so in my particular course, I try to show how we can take all that math and some of that physics and science background and how we can apply that actually in a chemical engineering situation for a plant. And then the other course I teach is biomass energy systems. And so it's really close to my research. Um, really, the course really is a technology survey. We talk about different types of technologies to make biofuels. But it's not just the technology component of that I think is pretty interesting. It's all the things that's surrounding it, right? So we can have the best technology, but if the economics aren't right, if people aren't going to use it, if it's not environmentally sustainable, these types of things, if you don't consider these types of issues, these technologies won't ever be commercialized. And so that brings me to my research interest and to the title of the presentation today, which is the production of fuels, chemicals, and materials from plant biomass specifically. So what I'm really interested in is how do we take plant material, woods, grasses, those types of things, and how do we produce fuels? Not just fuels though, but chemicals and materials. And I say this is important because if you look at the petroleum industry, right, and you think about what is actually produced from crude oil. Not only do we get gasoline and diesel and jet fuel from crude oil, but just about every type of commodity chemical that you see in almost any type of consumer good. So like your shampoo that you use this morning, your soaps, all those types of things, all the chemicals that are in those things are derived from petroleum. And then just about every type of plastic, so the nylons, the polyesters that are in your clothes, that came from crude oil. Um, and it's not just the fact that you are able to make this wide distribution of products, but that some of the products have a lot of value, right? So think about Kevlar. Kevlar is the material that's in bulletproof vests. And the money that they can extract from the same amount of carbon that goes into gasoline, they can extract out 100 times, 1,000 times the value for that particular carbon. And so if we are ever to generate an industry, a bio-based industry, where we produce the majority of our transportation fuels and materials from plant biomass, we're also going to need this product diversity and this value-added product production. And so, some of the projects that I'm working on right now is how do we take, say, a plant material, and how do we generate a plastic that can conduct electricity from that biomass. Another project is how do we take plant biomass and then make a high strength polymer. So a polymer that has some of the same properties as, let's say, steel does, but is much lower lightweight, right? So that's essentially what Kevlar is. It's a high strength polymer, right? Um, and so the presentation that I give today, or the talk that I give, unlocking the secrets to plant cell wall or affordable ethanol. It was some work that I did as a postdoc and research scientist within the Bioenergy Science Center, funded by the DOE, Department of Energy. It's about a $25 million a year organization, right? And I always like to tell this story because a lot of people just have no clue where their tax dollars are going, right? And so these are some of the great things that the government's doing with your money. They collected about 300 to 400 different scientists, research scientists, principal investigators, professors, graduate students, postdocs, technicians, and their single goal is to commercially produce affordable bioethanol to try to displace or replace current petroleum production. We know what the barriers are to economical and sustainable production of the bioethanol, and so these group of 300 people were focused on solving those particular problems. And so I'll give you a little short story today about one of the projects. I worked on a bunch of different projects within this funding agency. I give you one little short story about that project, but more in general, I wanted to give you a little background on the motivations for doing biofuels research and give you some basic terminology and some basic science behind biofuels. Okay. So, what is a biorenewable resource? Well, if you talk about renewable fuels or renewable resource, basically it's something that can be sustainably produced. It implies that that resource will renew itself on a rate such that it's available for future generations, right? And so this is a reason why petroleum is not a renewable resource. It takes hundreds of thousands or even millions of years to generate the crude oil that we extract from the earth. But for the most part, crude oil and biofuels are very, very similar in the sense that they have the same feedstock, right? So we use biomass, which can be widely defined as any kind of biological organic material. Um, um, when I talk about biomass, I'm typically talking about plants, but you can define algae, even animals as types of biomass. And so, in the production of petroleum, this biomass gets buried underground for hundreds of thousands of years under heat and pressure, and over that long period of time, it generates crude oil. And so, when we're trying to make biofuels, we're trying to replicate that process, but on a scale such that it's renewable. One of the great things about plants is that they do photosynthesis, right? And so, photosynthesis, you have light, CO2, and that plant has the mechanisms to sequester some of that light energy and actually store it as chemical potential energy. So if you don't know what chemical potential energy is, not what exactly that term means, you don't need to know what it means. It says we're going to take the sunlight and we're actually going to make molecules. These molecules have energy that's associated with it, right? So we can take these molecules and then we can burn them. We can actually extract that energy out later on. And that's essentially what we do when you burn gasoline, right? Well, plants can take the solar energy and convert that into essentially carbohydrates and oxygen. And we take those carbohydrates or the material that the plants produce and we try to generate fuels and material. The photosynthesis that plants do is actually not very, very efficient. If you look at the amount of solar energy that the Earth is exposed to, it only sequesters about 0.02% of that energy. But even if you took that energy, it's about 40 gigawatts, right? Which is about 3 to 4 times the amount of the world's energy usage. The important part here is that obviously we couldn't recover the entire 200 billion metric tons of biomass that is produced every year and actually convert that into biomass. Into fuels. That wouldn't actually be feasible. But I bring this point up is because there is a really abundant source, if you look at biomass for something like trying to displace fuel and chemical production, global fuel and chemical production. So just a few things about the motivations for researching biofuels. Essentially, petroleum is a finite resource. A approximately 47% of the total recoverable reserves of conventional oil have already been extracted from the earth. Now, this is kind of a touchy subject because what I specifically said was recoverable conventional oils, right? And every day they're coming up with new technologies to pull out, different types of oils. Some of these oils aren't conventional, so like the tar sands that people talk about that's coming from Canada, and they want to send through the pipelines. That's not a conventional oil, right? And it actually requires specific types of processing to actually extract that oil out. There's probably lots of uncovered oil and oil fields under Antarctica and the Arctic that we just have no clue how to get to or even that they're there. But the point that I'm trying to make here is that of the recoverable oils that we do know that are out there, of these conventional oils, we've consumed approximately half of that. And half of that consumption happened since the turn of the century. So if you look at the speed at which we're consuming oil, if you just plotted that out, we will run out of oil reserves in like the next 50 or 60 years. Now, of course, this is all predicated on this idea that there's probably tons more of oil that's actually out there, and there's a lot of oil that I'm not defining as conventional oil sources or recoverable oil sources. Environmental quality, everybody knows about issues with aerosols, so like the particles that get released during combustion, CO2, those types of things. National security, during the 1970s, there was an artificial increase in oil prices. This was a consortium of people in countries in the Middle East. They wanted to exert control over certain political control over other countries. They artificially caused a shortage. And since then, the United States has been very, very invested in decoupling our economy from some of these other foreign cartels. Access to increase agricultural production and improve rural development. All of these are reasons for generation of biofuels. If you look at the electricity mix, if you look at it here, most of our electricity, this is in 2005, is generated from coal, and only a small amount comes from biomass. So there's a huge area here that we can expand into. Similarly, for transportation fuels, this plot might be kind of hard to read, but essentially, this little small area here represents biodiesel. This little small area represents ethanol. Asia, North America, Europe. But the point here is that the majority of our transportation fuels, again, they don't come from biofuels. So let's just talk about some of the different types of biofuels. So the biofuels that we define as first generation biofuels are the biofuels that we use today. And they occupy three major areas: corn-based ethanol, vegetable biodiesel, and what we would call biogas that was generated from anaerobic digestion, right? And so most people are familiar with the corn-based ethanol. And so what they do is they grow the corn. Corn has a lot of easily recoverable sugars, and so it's a great source for the production of ethanol. They recover those sugars. They ferment those sugars into ethanol. It's a really old process. That's how monks made beer, and I don't know, long time ago. But we take that process to use it to produce ethanol. And there's a lot of problems with these first generation ethanol sources. For the most part, none of them have high yields and are very economically competitive. And so they all, for the most part, require heavy subsidizing from the government. These numbers are a little bit old, but for this first generation biofuel, the production cost is somewhere between $1.85 a gallon to about $2 a gallon. And that includes feedstock and transportation and actual production separation of the ethanol, whereas the cost of production for gasoline is somewhere around 20 cents. Now that's the cost for the refineries. Now the feedstock itself is highly dependent on the cost of the oil itself. So at about 100, I think the cost of oil right now is about $116 a barrel. So that accounts to, if you look at per gallon cost, that's around $2 a gallon, right? So in general, we can make bioethanol on a cost level that's very competitive with that of petroleum. Now, the problem comes in is that we're talking about the feedstock being being $116 a barrel, right? Well, in 2005, that cost was $30 a barrel. In 1960, that was $2 a barrel. There's huge fluctuations that occur with the cost of petroleum, and it has a lot of factors that they can't predict, right? And so, if I'm an investor and you say, okay, I need $100 million to build a huge bioethanol plant, we're not quite sure what production rates we can do and specifically at what cost, because this is the first plant that we're building. As we do more and more plants and as we operate this plant for longer and longer times, we get better at predicting this and actually narrowing this cost down. But for the first 10 years or so, we're going to have to give you really, really the high-end estimates of how much cost and it's going to be associated with it. And even with that, we can't predict what the cost of oil is going to be. If the cost of oil, for whatever reason, drops down to $20 a barrel, then the rate of return, or how long it takes for you to make your money back, may go from 10 years to 20 years to 30 years. Nobody's going to make that investment, particularly when they can just buy stocks in an oil company, right? That's making like millions of dollars a year. Similar for the cost of vegetable oil biodiesel. I think that's somewhere between, depending on the source, between $1.30 and $2 a gallon. And again, the cost for petroleum-based diesels is approximately the same. So if we talk about second generation biofuels, this is what I actually do my research in, right? And so what we've done is we ferment the sugars to produce ethanol. These sugars come from the wood or grass, right? So within wood and grass, there's a polysaccharide that's called cellulose. It's just a bunch of glucose monomers, glucose compounds chained together in a big, long molecule. But they're hard to get at. The plant itself was specifically designed over evolution to resist the organisms or the enzymes that we try to use to break down the cellulose, right? So essentially what we do is we get an enzyme from a fungus, and we use that enzyme to try to break down the cellulose into sugars, right? But the plant, over millions of years of evolution, was specifically designed to resist that fungus, because that fungus and those plants exist for millions of years together, right? And so its very structure was designed to keep us, and I say us, but as humans, and utilizing these enzymes, these microorganisms, away from the cellulose. And so before that process, we have to do a pre-treatment, which is essentially we just cook the wood. It makes it easier it opens up the structure of the wood or the grass and allows the enzymes in. And then before that, we actually have to size reduce the wood, and then we have to grow and produce and transport it, right? So that all accounts for the cost. And what we've done is we've identified this step where the enzyme tries to break down the cellulose. We call that recalcitrance, the ability of the plant cell wall to resist deconstruction. So this is a SAT word for you, recalcitrance, right? You can use it to recalcitrate. You can use that word to actually describe a person. People can be recalcitrant, actually, right? So, but we're using it to describe the wood. So it's the plant cell wall is being recalcitrant to the ability to deconstruct it, and we define this as being the major hurdle to affordable ethanol. All right, and this is what is considered second generation biofuels, or the fuels of tomorrow. A lot of other people may have heard of algae as a production source, so these are called third generation biofuels. And from algae can actually produce lipids, which are essentially, that's how you make biodiesel. So lipid is a fatty, is the same stuff that's in fats for the most part, right? And so we can use that lipids to produce biodiesel. Also, algae accumulate cellulose. Again, this is a sugar that we can it's this polysaccharide that we can break down into sugars that can produce ethanol, right? And so there's a lot of advantages to using algae. They grow fast, they absorb CO2, they can be grown on wastewater, but it's really expensive, and because we have to use large areas of land, And more than that, it's really hard for us to engineer algae, right? Because it's in an open pond. And so any engineered system is going to be weaker than the natural system, right? So evolution has found the most stable microorganism to do a process, right? And so we engineer that microorganism. We're essentially making it weaker than the natural version, right? And so in an open pond, you never have these engineered algae survive. And then fourth generation biofuels, which are again these are the fuels of the future. We use engineered plants and we use engineered microbial systems. What is a microbe? Well, a microbe is essentially anything that you can see in a microscope that sees bacteria, fungi, protozoa, those types of things. And what we do is we engineer the plant to make it less recalcitrant, and we engineer the microbes to make a more efficient enzyme, or to take that enzyme and to take the number of steps and reduce them, right? So instead of having a step, we have an enzyme treatment and then we have a fermentation. Why don't we just engineer a microorganism that can produce the enzyme for us right there? So it's the enzyme producer, and it can use the sugars that are produced from that deconstruction of the cellulose and do fermentation. And so these were the types of projects I actually worked on. And so what I described mainly is a biochemical route to produce bioethanol. But there's also thermochemical routes. Thermochemical routes will use heat, pressure, catalyst. And so I am not a biologist by any means. I don't do metabolic engineering or anything like that. Microbiology. I'm actually a chemist that's in a material sciences body. And so I actually do a lot of thermochemical work right now. And what I'm really interested in is how do we blend thermochemical routes with biochemical routes. And this just kind of is a picture of what the cell wall looks like. And so this is a plant cell wall, and it's really, really complex, right? And so you say, well, why is it so difficult for us to understand how to break down the cell wall to produce these sugars that we then ferment into ethanol? Well, because the plant cell wall is this heterogeneous material, so that means it's different throughout the material itself. It's really, really complex, and it's multi-scale. We have order. We have the plant has order on the angstrom level, so individual molecules. The plant knows how to specifically place those. It knows how to specifically place the molecules themselves, so how all the polymers are placed together. And then there's larger anatomy structures on the plant that actually help defend it as well, right? So the way the morphology of the leaves, the way the leaves are, the types of pores that are on the leaves, all these things are designed specifically to make it difficult to deconstruct. But for the most part, I mainly worry about this molecular level organization. And so you have these glucose chains. Again, glucose is just a simple sugar, and these things are put together in a chain. So this is what a cellulose chain might look like. And then these cellulose chains are then packed into fibrils. And so this is a cellulose microfibril. And then these microfibrils are packed within the cell wall inside of a matrix. And they are wrapped with other polymers, primarily two other polymers. One is a hemicellulose, which is very similar to cellulose, but it's made out of a bunch of different types of sugars, not just glucose. And it's really, it doesn't form these really strong fibril structures. And then you have something else called lignin, which is another molecule that gives the polymer, that gives the cell wall strength. So this is what a hemicellulose chain might look like. This is what a lignin chain might look like. And this is, again, the structure of the cellulose. And so, again, I just want to go back to this idea of what the Bioenergy Science Center is all about. It's $25 million. It's completely dedicated to understanding the structure of the cell wall and then trying to overcome its recalcitrance, right? And the reason why I bring this up is because I got to work with microbiologists, synthetic biologists, metabolic engineers. I got to see how big science was done. It was really exciting. I got to travel a lot. It was one of the greatest experiences of my life. And if you guys are ever go to grad school or something like that, these are the types of projects you want to be part of. And the way they plan on doing this, and the way that I think this problem is going to be solved, again, is that they do genetic engineering of plants. So the idea is like, okay, well, let's try to figure out genetically why these plants are so difficult to break down, and then let's try to re-engineer them. They not only try to re-engineer them to make them easier to break down, but to have faster growth rates, higher yields, to resist all types of pests. to be more water, to use less water and fertilizer, all these things that would be important if we were going to try to displace large amounts of petroleum with biomass, right? Then there's this area of improving enabling technologies. This is where I actually came in, right? And so we talk about pre-treatment of the biomass to make it easier to break down. We talk about separation technology. All of these things are really, really important to making a cheap ethanol product. And so we have people working in this. But then also we just need to have the ability to analyze these systems, right? It's like, how do we, as researchers, understand what's the best structure for a plant to be broken down? What's actually occurring? What are the mechanisms that occur during the deconstruction? And so we actually, a lot of this technology is a material characterization was specifically designed for synthetic systems. And so what we want to do is figure out exactly how do we apply techniques or even make new techniques to specifically answer the questions that we want. And then lastly, engineering biomass deconstruction. This is actually really, really interesting. If I ever had to do it over again, I would have totally done synthetic biology and metabolic engineering. Well, that's some of the coolest stuff I've ever seen. But these guys, they tinker with microorganisms and proteins, and they figure out what's the best systems to actually turn the glucose, the cellulose, into ethanol. Okay, so I'm just gonna, I got like 3 more slides here that I'm not gonna, unless you guys want to hear more about like specific projects or anything, I'm not gonna like super bore you with like some of the details of my research. But one of the coolest projects we worked on was this genetically engineered grass. So switchgrass is a high potential dedicated energy crop, right? So they're developing switchgrass as something that we can just put out in the big fields, and and its design is going to be specific for the production of biofuels, right? And so remember I was telling you the plant cell wall has 3 different components. It has hemicellulose, cellulose, and lignin fraction. The hemicellulose and cellulose fraction are both polysaccharides that can be broken down into sugars and then fermented into ethanol. But then we have this other component, which is this lignin, and it's really just like tough molecule, right? If we made this molecule synthetically, it would end up being used for some really high performance applications. It gives the plant cell wall its strength. It gives it its structural rigidity. It helps control water transport and a bunch of different functions. And so the idea is that, okay, well, if we have an enzyme, the enzyme attaches to the cellulose and it starts breaking off sugar molecules, right? Well, it can't do that if it can't get to the cellulose chain itself. And you saw how everything was wrapped, how the structure of the cell wall was all wrapped together, and that's part of its defense mechanism. Well, we thought if we could genetically engineer these switchgrasses to still be viable plants, yet to lower the amount of lignin in the cell wall, and maybe even change the actual structure of the lignin, right? And so this is a chemical pathway, biosynthetic pathway for the production of lignin, for some of the production of the lignin molecules. And actually, this changes like every 2 years. Like they don't actually, it's really hard to actually come up with these enzymatic pathways. They have some methods to try to determine things, but for the most part, they know that these enzymes exist and that they, in some way, do some kind of chemistry. So this is an enzyme. This is a C4H enzyme. We add this enzyme to this particular substrate, and it'll make this product, right? And so you can just see how the flux or the flow of the molecule would go until we get to a lignin structure. So this will represent a lignin, a certain type of lignin. And what we were able to do is like we know what the genes are for the production of this enzyme, and we can either turn that gene off so it doesn't produce this enzyme anymore, or we can ratchet it up and have it overexpress that, right? And so it'll force the flux through that particular pathway. Well, anyway, they actually engineered a bunch of different enzymes, or a bunch of different plants that had overexpression and knockouts in all of these pathways. But the one I focused on was this COMT, and they were able to downregulate this. And we thought, okay, if they can downregulate this or almost turn it off, maybe we want to get less lignin, and we're going to get less what they call S-lignin, right? And sure enough, that's what happened. So we grew the grass, and this is we had controls and we had different transgenic lines. And I'm going to try to explain why we have various transgenic lines, but the basic thing is I was able to run some characterization and determine the amount of lignin in the cell wall. And so when you go from the control to this transgenic line, you can see that the amount of lignin by a certain technique went from 200 milligrams per gram of plant to about 170, right? So we reduced the amount of lignin in the cell wall. Not only that, but the major monomers that make up the lignin, is what we call it H2 So where what I mean by a monomer is like the glucose is a monomer unit for cellulose. So in lignin, it also has monomer units. It actually has three different monomer units. Where cellulose has one glucose. And we were able not only to reduce the amount of lignin, but we were able to shift the distribution of monomers within that lignin. Right? You say, well, why is that important? Well, basically, what we were able to come out of this was that we used a certain technique, and this is all blurry, we have a lot of numbers, we don't even know what to worry about. This is the control, and this is the transgenic. And this represents the amount of sugar we were able to extract out of the cell wall, right? And so as we look to our transgenic, even without pretreatment, we were able to increase the amount of sugar that we can extract out of the wall using enzymatic hydrolysis technique. There's also a technique where we put enzymes, maybe all of the same way, and in that process, we produce directly ethanol from the cell wall. And in that case, again, we see an increase in the amount of ethanol that's produced. And I think the amount was specifically of 130% increase in ethanol production just from looking at these transgenic lines, right? And so this is just one transgenic line. They actually have been able to identify like hundreds and thousands of transgenic lines. And what they do is they actually run this through a pipeline, a high throughput pipeline to assess it. And then what they would do is then they would give me high potential candidates and say, hey, this looks like a good plant to grow for bioethanol production. Tell us why. And so that's what I did in that project for about 2 years. Just the last thing that I wanted to talk about was just the kind of things that if you're really, really interested in biofuels research, it's really an interdisciplinary deal, right? Pretty much from now on, all big science questions are going to be determined by interdisciplinary research, right? And so obviously there's chemical engineering that's involved. If you figure something out on a bench scale, it doesn't help unless you can actually process, hundreds of millions of pounds a year, right? And so you have to understand how that reaction changes as we go to larger and larger scales, right? And so, I don't know if you ever boil water, but the way when you're boiling water in a small pot, the time it takes and how hot certain parts of the boiler gets, changes as you go to bigger and bigger scales, right? And that's because you have these heat and mass transfer effects that occur, right? And understanding that is going to be huge. You got to understand bioengineering. And as I said, synthetic biology, metabolic engineering. If you don't know what it is, just keep it in the back of your mind. Someday, try ask one of your professors about it. Agriculture, agronomics. If we make a great grass that will be good for biofuels, we need to really understand what's the best way of planting that in fields and and stuff like that. Computer science. You say, well, where does computer science fit into this? These systems are so complex that we rarely can understand them without using some type of computer simulation, right? The way they actually identify the genes that we want to attack to or change or underexpress, overexpress for the reduced recalcitrance, sometimes they say, okay, well, we know that this gene codes for this one particular feature in the plant cell wall. And sometimes what they do is they just sequence the whole plant, and then they look at the genetic sequences, and then they compare it to other genetic sequences that they know from other organisms, and they say, well, in this other organism, it codes this genetic sequence codes for this particular feature, and in the plant of interest, we have a 70% or 75% or 85% match to that particular genetic code. Now we've identified a place to do genetic engineering. Biology, obviously, omics is really cool stuff too. Chemistry, and so my background is actually in catalysis, but I haven't done much of it until recently. And then material science, I do a lot of material characterization as well. So I'd like to thank you for your time, and if you have any questions, feel free to ask.
Okay, so we've got a mic here, so.
Are there any I've done some reading about stuff that
Mhm.
products for biofuels other than ethanol from cellulose and other feedstocks. So not biodiesel, not 16 carbon long, but more like 5. Levulinic acid, I think, was one that was brought up in the research that I've done. Um, I would think that'd be better because there's more energy in there than little 2-carbon ethanol. But what other stuff like that are you looking at with these processes?
So, actually, for fermentation processes, um, alcohol is some of the easier things that they produce, right? Theoretically, using synthetic biology, so the process of synthetic biology is saying we can literally just make whatever we want using a microbe, right? We can, if we have the enzymes, if we know the enzymes, we know the genetic codes to make those enzymes, we should be able to make whatever we want. But in general, we try to make compounds that natural molecules already make. And so alcohols are one of them. They do make larger molecular weight alcohol, so propanol and butanol and those types of things can be made from fermentation. As you go to these larger alcohols, though, you start to have huge problems with inhibition. So they don't naturally like to make propanol. They don't like to make butanol, right? And so they see the very product that we're trying to get them to make as a toxic compound. And so overcoming that particular problem for some of these larger alcohols is a problem. You can also get them to make things like levulinic acid and stuff. One thing about levulinic acid, HMF, which is hydroxy methylfurol, there's a couple other products that you can produce. Those are way more easily produced thermochemically. So we can take sugar in the presence of an acid and we call certain hybridization reactions, dehydration reactions, and we can make a long, wide range of those, right? And then the question is, what's the cost that's associated with these catalysts? What can we actually produce? What can we actually utilize, let's say, levulinic acid for? You can actually never use it as a fuel because there's too much oxygen in it. Um, it does have a decent heating content, but you can never use it as a fuel. But you can actually take levulinic acid, do deoxygenation reaction, and that's produce a fuel. And then you're adding another step in there, right? So these are the questions that we struggle with. Like, what's this nice balance between cost and productivity?
With the switchgrass, how would you keep it, if you're going to grow fields of it, how would you keep it from crossing over to other um, different types of grass to keep that from crossbreeding, I guess would be the term, but so that you don't infringe on other people's, like a sod farm or something like that?
Yeah. So that's a huge deal with like genetically modified plants, you know. And so they don't actually have a specific way of like once we put a genetically modified organism out into the world, they don't have a way to actually control that. Once it's out there, there's nothing you can do. It's going to produce pollen, spores, and all kind of stuff, and it's just nothing you can do to actually sequester that. And I'm sure people like Monsanto pour tons of money in to try to understand that process and see if there's ways of actually controlling that. I think the thought process though would be that genetic modification of plants has actually been done for as long as we've been able to cross, right? And the very fact that we are using synthetic biology and these types of new techniques to do genetic engineering is actually not that big a deal, right? And so, I mean, maybe you're worried about these material out into nature, but essentially what we look at is that we just set up evolution. It might have actually found its notes in anyway. Any other questions?
One of the other things that you mentioned as a byproduct were long chain polymers. Are these long chain polymers better? I mean, there's a lot of controversy right now over like BPA and other sorts of plastics being estrogenic. And are these equally less than, are they better plastics or not, basically, for these polymers?
So, I mean, that's kind of a loaded question, right? And so, like, let's say for example, BPA, right? For the most part, BPA are good. People have freaked out about it a little bit. Which I totally understand. You have your family, you kid out of a bottle that's made out of something that could theoretically reach the material. But for the most part, they're fairly safe. You think, I mean, they do a lot of testing and try to understand that the effect of this. Um, the products that we would produce obviously start from biomass. Theoretically, would be more biocompatible than synthetic-based polymers. However, we do a lot of weird chemistry to them. A lot of stuff gets done to them, but they're not in a native structure. And you can always see 20 years down the line, 30 years down the line, somebody finding out that the stuff that we're making right now that's going to be safer, and those definitely not safer. So that's why I say it's kind of a loaded question because for the most part, people in science and engineering areas they do their due diligence, and we know that there's a certain risk associated with anything that you do. Right, you go out in your car, you can do a car crash, right? So we use polymers that theoretically